Welcome to the video on enzymes. In 10th grade, or I guess it was 9th grade, you guys learned a lot about enzymes, so hopefully a lot of this is a review for what you guys learned in 9th grade. But here we go. So enzymes are proteins. So the important thing, the fact that they're proteins, is that they form 3D shapes, just like we talked about last week, about how proteins fold. And the job of enzymes is to speed up reactions. Okay. Now, one thing that's important to realize is that they speed up reactions that are spontaneous. And what that means is that the reaction will occur without the enzyme, but it just occurs extremely, extremely slowly without the enzyme. It eventually will occur, but extremely slowly. An example of this, to some extent, is hydrogen peroxide. So, if you leave hydrogen peroxide out in the sun, or just out in general, it will eventually decay into water. That will happen, but it will take years. There are enzymes which break down hydrogen peroxide. You guys are going to use one next week, and they can do it in seconds. So by itself, hydrogen peroxide takes about a year or two, depending upon the conditions, to decay into water. Whereas with an enzyme, the enzyme can do it in seconds. Now, enzymes speed up chemical reactions by lowering something known as the activation energy. And the activation energy here is the energy needed to start a reaction. Without overcoming the energy, the activation energy, a reaction cannot occur. Usually it's the energy needed to make the molecules collide, enough energy to make the reaction occur. It's like a little hump that the reactants must overcome in order to get to the reaction to occur. This is a graph. This is an energy diagram. We looked at energy diagrams a lot in chemistry. This is a energy diagram for an exothermic reaction, and the reason why that is that we have the energy of the reactants is here, and here the energy of the products is lower. So what happens here is between here and here, because it's lower, energy was released. And as a result, this is an exothermic reaction. If it was an endergonic reaction, the products would be higher than the reactants. They would be up here. But what's more important about this diagram is the hump. This hump here, from the distance from the reactants to the products, is known as the activation energy. And once again, it's the energy needed to start the reaction. Look at the color of the graphs, and I'm sure you don't have color in your handout, but you can point it out. The larger hump is the activation energy without the enzyme, or the energy that needs to happen, okay, for the reaction to occur. But the black one is without the enzyme. Notice the hump is higher. The red one is the activation energy, or the red hump is the activation energy with the enzyme. And notice that hump is lower. So here, in this case, what happened is the enzyme lowered the activation energy. Notice, though, in between the two graphs, the one without the enzyme and the one with the enzyme, so without and with, everything is the same. The energy of the reactants is the same. The energy of the products is the same. The only thing that's different is the height of the hump. And what the enzyme does in this case is lower the activation energy. Now what we need to talk about, and we're going to talk about it actually in a little bit more simplistic terms than you would talk about in a heavy biochemistry course, is how, specifically, enzymes lower the activation energy. So the question obviously now is how enzymes do this, and there are two methods. There are some other ones that are a little bit more complicated that if you ever take a biochem course and learn about sort of enzymes reactions, you'll learn more about sort of the movement of electrons, and it's kind of crazy. But enzymes do this by doing two things. The first thing that they do, and we did talk about this one a little bit in 9th grade bio, is that they bring the reactants together and position them so that they can react correctly. If you think about sort of like two reactants in your body, they're floating around in your cell. Okay, and in order to react, they need to meet each other and also meet each other in the correct position. Okay, what an enzyme does is an enzyme brings the substrates together so that they can react. And by doing this, it lowers the activation energy. The enzyme will like float around in the cell, find one substrate, find the other substrate, and put them together so that they can react. And that lowers the amount of energy needed to do the reaction. If there was no enzyme, there's going to have to be a lot of energy exerted for the two substrates to even find each other. The second thing that the enzyme does is it stresses or breaks critical bonds And we're going to sort of take a look at this in a second, but there are certain bonds in a reaction that need to break, and the enzyme can put pressure or bond to that area so that it's easier or more energetically favorable for that bond to break. These two things which we're going to look at now both occur in the, some of you might remember this, occur in an area of the enzyme known as the active site. And again, enzymes speed up reactions that would occur anyway. This is the bottom statement says, but the enzyme just makes them occur much more faster. So, in terms of enzyme structure, or how the structure of enzymes helps them perform these reactions. So enzymes are proteins, and like we said before, what that means, why it's beneficial for them for proteins to be enzymes, is they form these globular, tough word to spell, globular 3D shapes. Here, in this picture, this is a ball and stick model of an enzyme. And here, these are all amino acids that are folding in certain ways to make the protein make this shape. Okay? Now, what's important here is that the enzymes, all enzymes, have an active site. Right? It's a groove. Where the substrate can fit in. Substrate is another word for reactants. Can fit into the active site. And in this active site here, the enzyme is going to bring the substrate and the enzyme, the two substrates together to do a reaction. It's in this location here, though, that because you're bringing the substrates together in this location, the activation energy is lowered. Equally important, though, is the idea that this shape, and we're going to talk about why this is in quotation marks in a second, of the active site is specific for specific molecules. So there's not one master enzyme in the earth that can break down or build anything. Each reaction that occurs in your body has a specific enzyme that does the reaction. There are thousands and thousands of enzymes. So for example, there's a specific enzyme that's used to break down starch. That enzyme that's used to break down starch is known as amylase. There's also enzymes to break down proteins, and those are called proteases. But there are specific enzymes to do the reactions in your body. To just simply make energy, to take sugar and turn it into energy, the process of respiration, you need about 13 to 20 different enzymes just to do that process. Take glucose and turn it into ATP. But the point is that it's the shape of the active site that makes this enzyme specific. In 10th grade, or not, sorry, 9th grade, we drew enzymes like this, and then we're like, which substrate will fit in the active site or something. That's what we drew. And we're like, And if you didn't really know anything about biology, you would have said, okay, well, this one looks like it fits. The whole idea of fitting thing, it's not necessarily shape that makes the enzyme specific. In 10th grade or 9th grade, we simplify it, saying it's the shape of the active site. It's actually that more of the chemical groups that fit together, which we're going to take a look at the next slide. So this looks like a complicated picture of an enzyme's active site. Yours is not colored like mine, so you may want to take a second and pause the video and color it a little bit. The red amino acids are part of the enzyme's active site, and then the blue is the substrate. And what the point of this is that it's not so much that the substrates fit in, which is true, but they also have chemical groups that allow the enzyme, the red part of it here, to actually bind to, either through covalent bonds or intermolecular forces such as hydrogen bonding or ionic interactions, that allow the substrates to actually bond to or bind to the enzyme's active site. So this chemical group here sort of is able to bind to the carbon here to fit the substrate in the active site. If this if this amino acid went away, then, for some mutation or for some reason, then this enzyme would not be able to work because this substrate would not be able to chemically bind to this enzyme in the active site. So it's more complicated than just the substrate fitting. It actually chemically bonds. So here there are chemical bonds or interactions in intermolecular forces that allow the substrate to fit in to the active site. So it's more complicated than just two puzzle pieces per se. There are actual chemical groups in the amino acid provided by in the enzyme provided by the different amino acids that speed up allow the substrate to bind and therefore speed up the reaction. If one of those amino acids that's in the active site is removed for a mutation, and we'll talk about how that happens later in the year, how that possibly happens, then this enzyme could be rendered useless because the enzyme will not fit. Sorry, the substrate will not fit in the active site, and then the reaction cannot occur. So this slide shows the model of how enzymes work. Here we have the two substrates. We're trying to do a dehydration synthesis reaction with them by joining them together. And the first thing that happens is the enzyme finds both of them and places them in the active site. Okay, and again, these substrates are held into the active site by IMFs. So hydrogen bonds, ionic bonds that keep them in the active site. Not so much that it fits in, but their bonds are there conformed so that the substrate can fit into the active site. This, when the enzyme and substrate are together, is called the enzyme substrate complex. The act of this being into the active site lowers the activation energy, or the EA, and then as a result, the substrates are converted into products, and the products are released. As a result, though, if you take a look here, the enzyme here has gone back and can recatalyze this reaction again. It can do an unlimited number of reactions. It doesn't just do one reaction and it's done. Another thing that's sort of important and is a small distinction here is that notice how the enzyme does change shape a little bit during the reaction. So when the substrates bind, it changes a little bit to better fit the substrate. And it changes a little bit, and then when the reaction is done, it goes back to its original form. That's sort of a change in the two models that we have of enzyme action, which we're going to discuss now. So the two models that we have for enzyme action that scientists propose are induce fit and lock and key. This was the first one that they proposed. And they proposed that the enzyme is like the lock and the substrate is like the key, and the substrate fits into the enzyme, or the key fits into the lock and opens it, or the reaction occurs. But the lock or the enzyme doesn't change shape. So they didn't think there was no changing shape here. No changing shape. It turned out that this model is wrong. What's more correct is the induced fit model. And what the induced fit model says, and I'm not going to write, you should write what I say here, it's a little practice. But the induced little practice with that, what the induced fit model says is that when the substrate binds, the enzyme changes shape slightly to better fit the substrates. Then it catalyzes the reaction, and catalyze actually is an important word. Catalyze is a word for speed up. Enzymes catalyze reactions. They use that word a lot. And so the enzyme slightly changes shape to better fit the substrate, catalyzes the reaction, and then goes back to its original shape. So unlike the lock and key, where the lock always stays the same, in induced fit, or the enzyme always stays the same, in induced fit, the enzyme slightly changes shape. So at this point, videos can only be 15 minutes long, and I'm approaching the end. So what I'm going to do is I'm going to stop this video, save it, and then I have about 3 minutes left, but I don't want to be stopped in the middle. So then do the last couple slides in a second video.